Yeah, this did not get funded. It did get scored. It's tough. The money's tight. They only awarded 4 R21s in this study section. So it's tight, but we'll see what happens. So I'm going to treat this as somewhat like a thesis prospectus defense. Because it's kind of funny. What I'm really trying to introduce to you is an argument for a study and an argument that you may want to give me some money to do the study with. In the title of this, I referenced the holy grail of matrix biology. Matrix refers to the extracellular matrix of tissue, the stuff that's in between the cells floating around the different tissues in your body. Now, this holy grail is my analogy. Upregulating the elastin gene may not indeed be the holy grail of people who investigate the extracellular matrix and treat matrix disorders, but certainly if we could do this, it would change the world. Elastic fibers are part of the extracellular matrix in the connective tissues throughout your body. So this includes the arterial walls in your circulatory system, it includes your skin, and it includes your vocal folds. Elastic fibers do exactly what their name suggests. That is, they impart the property of elasticity upon these different tissues in which they reside. Unfortunately for all of us in this room, our body has produced really all of the elastin that it's ever going to. The elastin gene is typically turned off after puberty. And this is one reason why aging is a visible phenomenon. For instance, we know that our age, or that our skin ages, we can see that. And one of the things that happens is that skin loses its elasticity. And part of the reason for that is that the elastic fibers that are in the extracellular matrix of your skin are becoming disorganized, and they're not being repaired well, and their new ones are not being created. So if only we could turn that elastin gene on. I want to acknowledge some collaborators in this line of research that I've been involved in for about 5 years now. These include Bernard Rousseau, who's at Vanderbilt University, Bob Meekum at Washington University in St. Louis, Brent Cooper here at TCU, who's over in the Department of Psychology, and Jeff Marler, who's at Ohio State University. You'll see their names pop up on some of the publications in this line of research. So the focus of this project centers on the tissue of the vocal folds and how changes to that tissue can influence the sound that the vocal folds produce. Now we know that genetics influences either the tissue of the vocal folds or how those vocal folds function. And there have been a number of reviews that have been done on the relationship between genetic disorders and voice production, and it's been shown that there are at least over 100 different genetic conditions where a voice abnormality is part of the phenotype, that is the presentation of the person, specific to that condition. So, although we know there's a strong relationship between genetic abnormalities and voice disturbances, we really don't know a whole lot about the underlying causes of voice changes in those conditions. Specifically, there have been very few studies which have investigated the effects of gene abnormality on the vocal fold tissue itself. The sound of your voice is a product of both a sound source, that is the vocal folds in the larynx, and the vocal tract filter, that is the spaces above the vocal folds, which resonate the sound that the vocal folds produce. Any disturbance in the tissue of that sound source, in the tissue of the vocal folds, either through muscular misuse, what you do to change the shape of that tissue, or in actual changes to the tissue itself, are going to affect the vocal sound or vocal function. And we know this through a history of clinical studies, which have investigated this in human populations. However, although changes to the voice are characteristic of many genetic disorders, we don't have scientific evidence which points to why in most, if not all, of these genetic conditions. Recently, the National Institute on Deafness and Other Communication Disorders released their strategic plan, ranging from 2009 to the current year. And I thought this was a beautiful plan. because selfishly it related directly to the line of research that I've been developing since about 2007. Specifically, priority area number 1 states a need to study common variations of human DNA and their impact on susceptibility to human communication disorders, and the development of animal model systems to study the function of specific disease types or specific genetic conditions. And, lucky for me, I've been heading in this exact direction with my research. So the development of the line of research that this project is based upon began with investigations into humans with a genetic condition called supravalvular aortic stenosis. In this disease, one of the 2 elastin genes, and we call a pair of genes alleles, so you have 2 alleles for any gene. One of these elastin genes is abnormal in some way, either due to a mutation in the gene, a translocation within the genetic code, or deletion of the genetic material that's part of the DNA. It results in a heterozygous abnormality, heterozygous meaning one side, one gene, that results in a non-functioning allele. So one elastin gene is not working. Instead of having 2, you only have 1. And when we have only one functional copy of a gene, that we call that a haploinsufficiency. So the underlying cause of SVAS is an elastin gene haploinsufficiency. Instead of having 2 normal genes to produce elastin, they only have 1. The primary pathophysiological consequence of elastin haploinsufficiency is stenosis in the ascending aorta, which I can point out right there. So that's stenotic, it's narrowed down. And this happens because the elastic fibers in that region are both low in number, they're decreased in number, and their structure is disorganized. And as a result, what happens is smooth muscle cells proliferate in this region, causing it to narrow down. And this is a life-threatening condition, so a number of individuals are going to need surgery to correct this, or at least treat this, just to save their life. Now, for me, the interest in this genetic disorder lies in the fact that there are numerous anecdotal clinical reports that note a voice disturbance as part of the clinical phenotype of this disease. A voice quality disturbance is also characteristic of a related condition called Williams syndrome. In Williams syndrome, the elastin gene, along with 20 other genes in the same region where the elastin gene is located, are abnormal. Because of the inclusion of many different genes in this condition, the phenotype of Williams syndrome is much more complex than SVAS. These individuals will probably have SVAS, but they're going to have a complex clinical picture that goes much more beyond just stenosis of the ascending aorta. So it at least does include the cardiovascular disease that we see in SVAS, which is due to the underlying elastin gene haploinsufficiency. Other gene abnormalities related to Williams syndrome have been linked to visuospatial problems and intellectual disability. They still haven't mapped all the genes to all the different clinical characteristics that they note, but they have mapped the elastin gene haploinsufficiency to the cardiovascular problems that these individuals have. Now, specific to voice production, individuals with Williams syndrome have been described as having a harsh or brassy voice, suggesting another clinical population where this genetic disease results in something at the level of the larynx and the vocal folds. Even in non-genetic conditions, abnormalities in elastic fibers can negatively affect voice production. The most common voice disorder in those who are age 65 and older, I won't call those the elderly, is the result of a normal process of aging, and it's called presbylaryngeus. In presbylaryngeus, the structure of the vocal folds change, and this can be seen on laryngeal endoscopy. The vocal folds begin to bow, and there's some atrophy going on within the vocal fold structure itself. And that alters the vibratory dynamics of the vocal folds, how they move, which changes the structure of the sound that they produce. Clinically, these presbylaryngeus presents as a very breathy voice with a loss of vocal power. Okay? And it's one of the reasons why when you're speaking to a very elderly person on the phone without even seeing them, and guess that they're an elderly person, just by the sound of their voice. So it's believed, or it's been theorized, that elastic fibers within the vocal folds are one of the primary underlying causes of the voice change in presbyopia. That if something's happening to the elastic fibers and the elasticity of the vocal folds, which affects how they vibrate and the resulting sound. However, there's no research that exists which directly relates structural changes in elastic fibers with the acoustic consequences of that change.